Assalamualaikum warahmatullahi wabarakatuh, and welcome to our video, which is for assignment under course CHM256. And me and my teammate had chosen a topic about drug testing with chromatography. So I hope you guys will enjoy on this video and will get more information about it. So first thing first, I will explain more briefly about my own teammate that we had been work together while preparing for this video presentation. And each of them had their own section to explain more briefly about this topic, which is drug testing with chromatography. So for the first one, we look at Muhammad AIMAN bin FAKHUL ZAMAN. He will explain about the methodology. Next, myself is Muhammad IRFAN bin HAZLAN, and I will explain about introduction. Next, Muhammad NAZMI NAQIB bin Muhammad NAQIB. He will explain about example on chromatography for drug testing. NOOR AFRINA HAZIRAH BINTI SALEH. She will explain on conclusion. And lastly, NURUL SHAHIRA NAJIB bin ABDUL. She will explain about theory of drug testing with chromatography. And this is the list of contents that has on this video. So now, without further ado, let's proceed with our lesson.
Assalamualaikum and hi, my name is Muhammad HAZLAN. So now I'm gonna explain about chromatography technique that we will use on drug test. So for introduction, I will explain firstly about what is it for a drug test. So drug test on my research is a test that looks for the presence or absence of an illicit, or we can know it as a legal substance that have on some person that using a biological sample such as blood and urine. And next, for the next one, we need to know first what is the meaning for chromatography. So chromatography is a technique in which the components of the mixtures are separated based on difference in the rates, because we need to know every material they have their own density. So that's why every material has their velocity or their rates differently. So now, on our previous slide, I had explained a definition of chromatography technique. So why this technique we can do and we can apply for drug test. So firstly, for this technique, as a highly sensitive that can detect drugs and their metabolites at very low concentration. So additionally, this technique can identify specific drugs. So this is essential for forensic analysis and drug testing applications. And this technique also can be used to quantify the amount of drugs and metabolite present in a sample, which is the sample from blood and urine on some person. And this is useful in therapeutic drug monitoring applications to monitor drug levels and determine the extent of substance abuse. So now we're going to look for the next slide.
Okay, next, this is the theory of chromatography. Before I move further, I would like to introduce myself first. My name is NURUL NAJIB binti ABDUL. One of the most important analytical chemistry techniques is chromatography. It was initially created in 1906 by M.S. Sweat in order to separate colored substances into their constituent parts. Chromatography facilitates the identification, purification and separation of constituents from a mixture. Two key ideas that clarify how the separation is achieved in chromatography are the plate and rate theories. The components of the sample are divided into distinct bands during the chromatography process, resulting in a peak in the reading. The topic deconstructs chromatography's rate and plate theory as well as the fundamental idea underlying the method. The basic principle of chromatography is Based on the concept of distribution of components of a mixture of organic compounds between two phases, which are stationary and mobile. This is the rate theory of chromatography. Chromatographic analysis relies heavily on the rate theory of chromatography. The theories help understand how the analytes move in the stationary phase as the mobile phase flow through it. The activity in a chromatography column is defined by the rate theory of chromatography. It demonstrates how the elution rate and the solutes impact on the band's shape are related when the solutes elutes from the columns. The Y and form of the elution bands as the mobile phase move through a column can be inferred from the rate theory. It facilitates comprehension of the band broadening process and contributing factor to peak dispersion. It provides a plausible account of what goes inside a chromatography. Okay, next, this is the plate theory of chromatography. The plate theory of chromatography is an antiquated idea that provides an unrealistic picture of column efficiency and column phenomena. In 1941, Martin and Singh incorporated the theory into the chromatography technique. The theoretical plates, which are distinct layers, make up the chromatographic column as per the model. These plates are fictitious zone or stage that support the creation of phase equilibrium. In contrast to the rate theory of chromatography, it provides a theoretical illustration of the analyte separation process in a chromatographic column. The sample separation equilibrium between the mobile and stationary phases is provided by these plates. As you can see, this is the process step. First, we have sample injection. The mixture to be examined is added to the chromatographic apparatus. Second, we have separation. As the mixture passes through the stationary phase, various chemicals move at different rates depending on how they interact with the stationary and mobile phases. Third, detection. As chemicals depart the stationary phase, they are identified by an appropriate detector. As example, we have UV detector and mass spectrometer. Last, we have data analysis. The detector's reaction is recorded as a chromatogram with peaks corresponding to various chemicals.
In this part of the video presentation, I will like to share with you all one or two principles of thin layer chromatography, or TLC for short, towards drug testing. To begin, thin layer chromatography, or TLC for short, is a method used in drug testing to separate drugs in the sample by analyzing the mobility in the silica gel. The polarity of the drug determines its mobility in the silica gel, which is the stationary phase, which is a highly polar due to its hydroxyl groups, enabling the silica gel plate to form hydrogen bonds with the drugs in the sample. The polar compounds have region of positive and negative charge, while non-polar compounds have an even charge distribution. Non-polar drugs are weakly bonded to the stationary phase, while polar drugs are strongly bonded to the stationary phase. As the mobile phase moves up to the plate, meaning moves with the mobile phase, non-polar drugs are more easily carried along, while polar drugs are strongly bonded to stationary phase, causing the polar drugs to move slowly compared to non-polar drugs. Next would be the techniques of TLC, or thin layer chromatography, used for drug testing. The first one would be the sample preparation, and in terms of extraction, the drug sample is typically dissolved in an appropriate solvent. So if the solvent or the sample is complex, such as a biological fluid, extraction steps must be used to isolate the drugs from the mixture. And secondly would be stationary phase preparation. In this part, TLC plates are usually made of glass, plastics, or aluminum and coated with a thin layer of stationary phase. typically silica gel, but we also sometimes can use alumina and cellulose or cellulose. And in some rare cases, pre-coated plates are often used for their convenience and reliability. Next would be the development, where a small amount of prepared slide is applied to the TLC plate using a capillary tube or microsyringe. The plate is placed in the development chamber containing the solvent or a mixture of solvent in terms of, in this case, would be the mobile phase. The choice of solvent depends on the properties of the drugs being tested. The solvent moves up to the plate by the capillary action, carrying the sample components with it. Different components travel at different rates, which this leads to the separation. Next would be the detection and visualization of the spots. Now, some of the spots may be invisible or colorless in the naked eye, that's why this type of method exists. Many drugs are visualized under the UV light because they're either naturally fluorescent or after being treated with the UV active region. Something like chemical sprays are sprayed with regions like iodine, anhydride, or sulfuric acid, which react with the sample component to produce color spots. Where in rare occasions also radioactive detection. In some cases, or in these cases, radio-labeled drugs are detected using autoradiography. So as you can see in the slide, I have provided some of the examples. Now we've reached to the final part, would be the analysis. Now there are three here which is first the RR value calculation. It is calculated by measuring the distance traveled by the drug spot and dividing it by the distance traveled by the solvent front. This helps identifying the drug spot, comparing it with the standard RR values. The second one would be densitometry. Now what is densitometry? It is when the intensity of the spot can be quantified using the device called densitometer, which measures the optical density of the spots. And finally, in terms of comparison with standards, the amount of drug in the sample can be found also by comparing the spot intensity with those of non-standards. And to finish the experiment or the activity, using a device like cameras, they develop plates. The developed plates can be photographed for records. And as for data reporting, the results including the RF values and any quantitative data are compiled into a report for future interpretations. This part will be the visualizing the substance spot equipment. Now we have two here. The first one will be UV light visualizing method, and the second will be the staining visualization method. So in summary, drug visualization involves using various equipment to identify and analyze substance spots. UV lamps, UV cabinets, UV boxes, cameras, and spraying cabinets, iodine vapors, spray tables, and TLC sprays, all of them are used to visualize substance spots. UV lamps emit UV light at different wavelengths, causing some drugs and compounds to appear as dark spots or bright spots on the TLC plate. Cabinets and viewing boxes provide a dark environment, while cameras capturing high-resolution images of the spots and their relative positions. Spraying cabinets apply chemical reagents to the TLC plate, creating visible spots or color changes, while iodine vapors can make organic compounds appear brown or yellow when exposed to the TLC plate. Spray box sprays, as I said, the device's name, sprays the developed TLC plate with the visualizing region, producing color spots or changes in the background. Next, TLC sprays move the sample components to different positions on the plate based on their affinity for the stationary phase and the mobile phase. To end it, the whole reason this type of method even exists because some drugs are colorless and invisible to the naked eye, making it essential to use these appropriate equipments and techniques to visualize substance spots. Now, this is a formula that we use for the analysis or calculating the RF value for this TLC towards drugs. So as you can see here, it is distance traveled by the solutes divided with the distance traveled by the solvent or the solvent draught. So here's one example of how the questions about the calculating the RF value could be asked during the test. So it says here, it has given that the distance traveled by the solvent front will be the 10 centimeters, while the drug spot is 6 centimeters. So based on the formula that I have showed you, we can use that formula and the values that we obtain to substitute those values into the formula. Then we shall get the answer of this question will be 0.6. Now in terms of interpretation, we can say that the drug spot has traveled 60% similar nearly to the solvent flux distance, and the 0.6 RF value also indicates that the compound has a moderate affinity to both stationary phase and the mobile phase.
And now, I will share with you guys some of the examples of our thin layer chromatography results. But first, I will show you guys a simple demonstration of how our thin layer chromatography, or in other words TLC, was being set up. Where in the video, you guys will see two diagrams prepared where it shows how our TLC is being done. Where in the diagram, you guys will see on the TLC, there will be the presence of tiny colored dots. These dots are the one that indicates the presence of chemical in our samples. That's why TLC are commonly used in testing the presence of drugs. I will proceed with our first example of our TLC results, which is the TLC of testing the presence of marijuana and hemp. Where you guys can see there's the photo which what will happen when there is the presence of the marijuana or the hemp. Next is the TLC of cannabis and tramadol, where just like before, the TLC will show and indicate if there are any presence of cannabis and tramadol in our sample. And lastly, the TLC of Navidar SP, where the TLC of Navidar SP is special because it is our daily life medicine such as paracetamol, and also it requires UV light or iodine stain for us to see the result clearly, because without the UV light or iodine stain, we can see the result with our naked eyes. Where with the presence of UV light and iodine stain, the result we will get if there are any presence of the drugs will be shown as the diagram prepared.
Hi, my name is AFRINA HAZIRAH binti SALEH. My ID number is 2022480046. For the conclusion, as we know, chromatography techniques including gas chromatography GC, liquid chromatography LC, and thin layer chromatography TLC are fundamental to modern drug testing. This method provides high sensitivity and specificity, enabling the precise detection and quantification of drugs and their metabolites in various biological matrices. Their versatility allows for the analysis of a broad range of substances, making them suitable for diverse applications such as clinical diagnostic, forensic investigation, and pharmaceutical quality control. Integration of chromatography with mass spectrometry (MS) enhance its analytical capabilities, offering detailed structural elucidation and confirmation of compounds. This combined approach ensures the accuracy and reliability necessary for critical application, from therapeutic drug monitoring to legal proceedings. In conclusion, chromatography techniques are indispensable for drug testing, offering robust, accurate, and comprehensive solutions. Their continued development and application will further enhance their role in ensuring public health and safety.